So the subject for today is a little bit of an odd one, but I want you to indulge me if you will. The topic is called Microchimerism, which is about the persistence of male DNA in the female body after intercourse. The term chimera originates from the Greek mythology, describing a creature composed of different animal parts, typically depicted as a lion with a goat's head and a serpent's tail. The concept represents an entity formed from multiple distinct organisms. Scientists have adopted this mythological term precisely because they wanted to talk about this biological phenomenon where an individual carries genetically distinct cells or DNA from another person. In the case of sexual microchimerism, male DNA may persist within a woman's body long after intercourse, akin to how different parts of the mythological chimera come from separate animals. Similarly, pregnancy-related fetal microchimerism also describes a long-term retention of fetal cells in a mother's body. The word microchimerism specifically denotes that these foreign DNA elements exist in small amounts within a host, distinguishing it from a broader case of chimerism where an organism contains large portions of another's genetic material within their body. So let's talk about microchimerism. This is a biological phenomenon in which foreign DNA or cells persist in an individual's body. While this phenomenon is most commonly associated with pregnancy, where fetal cells remain in the mother's body long after childbirth, there is a growing discussion around the possibility of sexual microchimerism, where male DNA from semen persists within a woman's body long after intercourse. This video will explore the potential that semen-derived DNA fragments can be absorbed by female reproductive tissues and the mechanisms by which male DNA may enter a woman's bloodstream and the possible implications of sexual microchimerism on the female body and the brain. So let's understand microchimerism. Microchimerism occurs when cells originate from another individual and they integrate into the host's tissues and persist over time, perhaps even decades. The most well-documented cases occur during pregnancy where fetal cells pass into the mother's bloodstream and can remain for decades. These fetal cells have been found in the maternal organs, including the brain, the liver, and the heart, potentially influencing immune responses and even tissue repair. Sexual microchimerism, on the other hand, suggests that male DNA from semen may be retained within a woman's body beyond conception. Though less studied than pregnancy-related microchimerism, this form of foreign DNA persistence could have significant biological implications, warranting further research. Let's talk about the composition of semen and DNA. So semen is a complex fluid containing sperm cells, proteins, extracellular vesicles, and various bioactive molecules. Sperm cells contain the male's genetic material and are the primary contributors to fertilization. However, semen also includes prostaglandins, hormone-like substances that modulate the female immune response, cytokines and growth factors. Molecules that facilitate cellular communication may also impact the female reproductive tract in this way. Extracellular vesicles, EVs, tiny membrane-bound particles containing RNA and proteins that can be absorbed by female tissues. The introduction of semen into the female reproductive system sets off a series of physiological interactions. Some components of semen are absorbed into the vaginal and uterine walls, potentially leading to the incorporation of male DNA into the female body. So how does male DNA enter a woman's bloodstream? Well, there are multiple pathways by which male DNA may persist within a woman's body well after sexual intercourse. There is absorption through vaginal and uterine tissues. The female reproductive tract is highly absorptive, allowing molecules and even cells to pass through its mucosal lining. Some sperm cell derived DNA fragments may be taken up by epithelial cells lining the cervix, the uterus, or even the fallopian tubes. Once inside, these fragments could integrate into local tissues or enter systemic circulation through the bloodstream. Interaction with immune cells. The female immune system interacts with semen upon exposure as it must balance immune tolerance to prevent an immune attack on sperm, with a defense against potential pathogens. Some male cells or DNA fragments may be phagocytized or engulfed by immune cells such as macrophages and dendritic cells. These immune cells can then transport the male DNA to other parts of the body, including lymph nodes as well as the bloodstream. Endometrial uptake and circulation. The endometrium, the inner lining of the uterus, undergoes cyclic changes that make it highly vascularized. During sexual activity and fertilization, semen components may enter the endometrial tissue and subsequently gain access to blood vessels, facilitating the movement of male DNA into systemic circulation. Evidence of sexual microchimerism. Although research on actual sexual microchimerism is not where we'd like it to be, some studies do provide some indirect evidence of male DNA lingering and being persistent within female bodies long after intercourse. The first is detection of male DNA in women without male offspring. Studies have identified male DNA in the brains and other tissues of women who have never given birth to sons. This suggests that sexual intercourse, rather than pregnancy, might actually be a source of male genetic material in these cases, since it did not originate from the fetus, because the fetuses in those cases were all female. Animal studies supporting semen absorption. Research on certain animal models have demonstrated that sperm-derived molecules in DNA can be incorporated into female tissues. These findings raise the possibility that similar mechanisms might occur in humans. Long-term presence of male cells in female organs. Some investigations have shown that male-derived cells or DNA can persist in a woman's body for years, potentially influencing immune responses and biological functions. How sperm-derived DNA fragments might also be absorbed by a female's tissues. The process by which sperm-derived DNA is absorbed and persists in female tissues may also involve several key mechanisms. The first is endocytosis by epithelial cells. Cells lining the reproductive tract can take up extracellular vesicles and free DNA fragments through endocytosis. Once internalized, these genetic materials may integrate into the cell's cytoplasm or even influence gene expression temporarily. Fusion with female cells. Some researchers propose that sperm cells that do not fertilize an egg may still merge with some female somatic cells, leading to the transfer of genetic material. This phenomenon, though rare, might allow male DNA to persist within female tissues. Integration into circulatory and lymphatic systems. DNA fragments or even intact male cells may enter the bloodstream or lymphatic system, where they can circulate and become embedded in various organs, potentially contributing to microchimerism. Male DNA persistence in the female brain. One of the most intriguing aspects of microchimerism is the presence of male DNA in the female brain. Studies have shown that male cells can cross the blood-brain barrier and become embedded in a woman's neural tissues, sometimes persisting for decades. This startling discovery raises important questions about the potential impact of sexual microchimerism on cognitive function as well as neurological health. What are the possible effects on brain function? Well, the incorporation of male DNA into the female brain could influence neurobiological processes. While the exact implications remain unknown, some scientists hypothesize that these foreign cells might play a role in actual neuroprotection and thus may contribute to some sort of behavioral changes. We also have association with some neurological changes that might become disorders. Some research has explored potential links between microchimeric cells and conditions such as Alzheimer's disease or the like. While fetal microchimerism has been associated with a reduced risk of neurological disorders, it is unclear whether sexually acquired male DNA has similar effects. Then there are further immune implications as well. The presence of male DNA in the female brain could alter certain immune responses, potentially affecting inflammation levels or susceptibility to neurological diseases. This area of research, though, is still in its infancy and has not a whole lot of meat to those potatoes, but nonetheless, the male DNA persistence may have more profound effects than previously understood. Potential biological implications of sexual microchimerism. The presence of male DNA in a woman's body could have several potential biological and physiological effects. The first is immune system modulation. The immune system may develop a form of tolerance or even reactivity to persistent male DNA, potentially affecting responses to subsequent pregnancies or infections. The influence on future pregnancies could be that some hypotheses may suggest that the exposure to semen from a long-term partner may improve reproductive outcomes with the same partner, possibly due to immune adaptation. Conversely, repeated exposure to different male DNA sources may have unknown effects on the success of future pregnancies. In conclusion, sexual microchimerism presents a fascinating but underexplored area of biology. The mechanisms by which male DNA persists in the female body, particularly in the brain, raise profound questions about its potential effects. As scientific understanding advances, sexual microchimerism may offer new insights into human biology, genetics, and evolutionary adaptations. Please don't forget to like, share, and subscribe. If you're new here, this is a safe space. If you're returning, I love and appreciate you so much, and I'm glad that you're back. In the name of the Most High God, I wish you health, wealth, prosperity, and abundance. Blessings and protection. Signing off. Take care. Have a great day.